Hello everyone, welcome to another lecture of drug delivery engineering and principles. We have been discussing about how to go about and use responsive systems. So, just a quick recap of what we learned in the last class. In the last class we started our discussion on various responsive delivery systems, particularly the temperature responsive delivery system. And so, what that entails? So, in temperature we first looked at LCST exhibiting polymers. So, this is nothing but lower critical solution temperature. And what this is typically you will find that most things become more soluble as you heat them up, whether it is some protein, whether it is some polymers, but there are some unique type of polymers that actually show the reverse property, where if you heat them up they become more hydrophobic and they actually become less soluble. So, such polymers are called LCST polymer, and the temperature at which this switch between the properties happen is called LCST. And what we found here that if you let us say make a hydrogel out of such polymers, which is let us say chemically cross linked, so all of these places are fixed, but the polymer itself will change property as the temperature changes. So, let us say if we now heat it up above the LCST. So, for example, let us say the LCST for this particular polymer system is about 30 degrees Celsius. So, at room temperature it is fairly well soluble, it is fairly hydrophilic. So, it will have a certain swelling ratio, it will interact with water molecules around it and will swell. But once you let us say go to 37 degrees Celsius, which is our body temperature, what will happen is this is going to become hydrophobic and it will not try to have interactions with water. So, it will then try to have interaction with other chains which are also hydrophobic. So, in that case what will happen is these polymer chains will basically repel water and try to come close to each other. However, since they are cross linked at these points, they cannot leave those points. So, there basically the pore size is going to shrink, and so that is what the LCST is, and we discussed how this can be done. Very similar thing, a variation of this is sol gel hydrogels, which are nothing but instead of being chemically cross linked they are physically cross linked. And that means that now these chains can move around. So, but then if they are becoming hydrophobic they may not actually form any kind of gel. So, what will happen in these cases is if you have a polymer, it is going to show a phasic diagram which is something like this, where you have concentration on the x axis and the temperature on the y axis. So, only within this area will this be gelled, anything above or below it will not gel. And so, that is because the chains will be too soluble at a higher temperature, but then if the temperature is in this range they will have enough hydrophobicity. So, actually do a physical cross link gel. Then we also talked about pH responsive gel, which we have been talking about throughout this course, but we looked at some more variations to that. So, how you can vary the pH and have more swelling or less swelling, and depending on that and other applications. Here we looked at an example where we had a colon specific delivery. So, we had polymer chains that were cross linked with an azo bond, and these polymer chains themselves had lots of COH group. So, in cases where this is the case, what will happen is as it is travelling through let us say the stomach, these polymer chains are non ionic because they are already protonated, their carboxyl is not going to lose its proton, and they have a certain pore size. So, maybe your drug is in here which is encapsulated. As they go to a higher pH in colon, now what is going to happen is these all are going to become charged. So, all of these will become charged. As a result, they will start to electrostatically repel each other and the pore size will increase. So, this is basically become like this, and not only that, there is a lot of microbiota in our colon which has azoreductase as an enzyme that is being produced, and that is going to cleave these bonds. So, eventually you will have your drug release in colon instead of your stomach or intestine. Then we also looked at glucose sensitive gels, and in these again there could be several variations, but one variation we looked at was a pore that was covered by polymers that were fairly water soluble. So, they will extend and not let anything within this device. So, let us say if your insulin is here, the insulin can't go there because these polymers are physically blocking these pores, but then lot of enzyme called GOD, glucose oxidase, was tagged to these polymers, and whenever enough glucose is present in the surrounding. So, if let us say a glucose come, this is going to react with this glucose oxidase and produce H plus ions, causing a local drop in the pH. These things will change property with the local drop of pH and will shrink down and essentially become confined to the surface rather than spreading out, hence opening the pore for the insulin to come out. So, these are some of the systems that we discussed in the last class, and then towards the very end we learned about some bioshielding strategies, and so those were more that let us say we have particles and we do not want our biological system, our immune system to detect them, then what is the way that we can prevent their detection, and there are several things that we can do as we have already discussed throughout this course, and this is just sort of a repeat of that. So, one thing that we can do is coat them with lipids. So, this is something very similar to your liposomes, which are actually made out of lipid, but you can take your polymeric particle and then you can coat it with these lipid layer, which could be made from these lipids that are derived from the human sources, and that will mean that the body will only see this and think that this is something of a self particle. Similarly, you can coat it with polymers. So, this is few things we talked about was PEG is one of the good polymers that people have been using, although we also talked about there are some antibodies that are being detected now against PEG. Similar to the PEG, we can use carbohydrates. So, this is again similar to what our normal cells are. Our cells have lots of proteins, and these proteins are all heavily glycosylated. So, they also act as a shielding layer. So, we can use something similar strategy on our particles, and then finally, we can coat them with proteins that are also present on the cell surface to sort of give a signal that this is a self protein or this is a self material. So, we will continue the discussion today in this class and talk about targeted drug delivery system. So, the targeted drug delivery system can now be divided into two categories. One is passive targeting, another is active targeting. So, passive targeting is what we have primarily talked about through this course now. EPR effect was one that was targeting to cancer as well as inflamed tissues. And the major rationale behind that was that the blood vessels that are present in this inflamed or cancerous tissue are much leakier than the healthy tissue. So, let us say if this is a healthy tissue and this is a tumor, then in these regions the blood vessels are leaky just because either they are immature or there is lot of inflammation that is causing this drop in the vascular network. So, what you can do is you can make particles that are big enough to come out through these pores, and so in that case they will keep on circulating through the healthy vessels because they can't go through, but as soon as they reach cancerous vessels they can come out. So, something like a particle which is 50 to 200 nanometer is targeted to the cancer. However, this is a passive targeting, because at this point you are not really forming any active bond, it is not very tumor specific, it is any tissue that is leaky in its blood vessel will cause accumulation of these particles. So, this is what we call as passive targeting. Other things are tumor environment again. So, you can have let us say your drugs or prodrugs which are targeted to let us say a local pH or something like that. So, that is again so let us say if I have a prodrug, so maybe I have this drug which is conjugated by some labile bond to this heavy molecule that is protecting this drug from its activity as well as its degradation, but then this cleaves at a low pH, and we know that tumor environments also have a low pH just because they have a very high metabolic rate, and so this drug will accumulate everywhere, but in the tumor environment it may break, it may also break in some other environments, and so this is how this is inherently again targeted to passively targeted to these tumor regions or any region which is low in pH. So, these are all good, but these are again all passive targeting, there is no active targeting involved, and obviously one other method is to use a direct local delivery. So, if it is for lung you directly give it through inhalation, if it is directly you want to deliver it to the tumor and you have a big tumor you can directly inject it into the tumor itself. So, that is also categorized in the passive targeting. So, let us look at what is active targeting, and as you can already see in the slide. So, we have this is specific to a direct interaction. So, let us say there is a receptor ligand. So, we know maybe a tumor cell because of its high metabolic rate and because of whatever mutation they might be expresses a protein X on the surface which is present only in the tumor or at least in much higher amounts in tumor. Then if I then put my particle with the ligand of X, then this is actively targeted to the tumor, because now this is going to go and bind to the tumor cells and the tumor microenvironment. So, that is active targeting. So, it could be on the basis of ligand and receptors. So, this particular example, this could also be that a certain lectin or a carbohydrate is present on the tumor and you conjugate your particle with the carbohydrate or the lectin to be able to target it to that tumor cell, or it could be any other cell, it does not have to be tumor, it could be other disease as well. And then finally, it could be an antibody antigen interaction. So, let us say we know that there is an antigen, this is very similar to the ligand receptor, but more specific in terms of its function and role. So, let us say if we know that there is an antigen that is present on the tumor regions or that is present on let us say in a bacterial cell or in mammalian infected mammalian cell, we can then take our particle, put these antibodies onto that, and these antibodies will then go and bind to this, and that will be more active targeting. So, that is the major difference between passive and active. So, there are lots and lots of targeted drug delivery systems that have been tried. So, this is just an example with liposome, but this could be true with any type of particle. So, here is just a normal non targeted particle, it is a liposome in this case, it is pegylated to have enhanced circulation, maybe its diameter is about 100 nanometer, and that is what allows it to accumulate in some regions. Maybe you have certain charge on this as well, maybe this is negatively charged, maybe this is positively charged, just depends on what your application and target is. And but if you are looking at targeted nanoparticle, what you will do is you can then modify this targeted nanoparticle in such a way that the exterior surface, whether it is the PEG chains or is just a particle with the exterior, is modified with some target or some ligand that is specific for the disease you are looking at. So, again there are several targeting moieties that one can go through. There are antibodies, you can use the full antibody. So, again antibody is also divided into an FC region and the variable region. FC region is basically the constant region. So, which means that this is fairly similar in most types of antibodies, and then variable region is what allows it to bind to different targets. So, this is fairly variable and recognizes different targets. So, sometimes since mostly for most targeting specific applications you may only need these regions. So, people are able to even separate out these segments to reduce the antibody size and not have anything else around. So, you can use antibody fragments as well. You can use any small molecule. So, maybe some cells are heavy in requirement for glucose. So, you can even put glucose on your particle and then may cause more and more receptors on the cell to bind to it. You can use a small peptide, again very similar concept. So, why reduce it to this, why can't we just reduce it to whatever the moiety of the peptide that is present on these surfaces that are responsible for its recognition. And then there is also another thing called aptamer, and we will talk about aptamer in next couple of slides. So, all of these are targeting moieties that are fairly used. So, antibodies, proteins, which are again antibodies is a type of a protein, you have lipoproteins, hormones, you can use charged molecules, and if you use charged molecule this is technically not an active targeting, this is passive targeting. You can use specific polysaccharides, some low molecular weight ligands, and again as it is shown pictorially here you can mix and match them, you can same particle can be put with 2 or 3 ligands to confer much more targeting to such particles. So, as I said aptamer is another class that I will just describe, it is not probably as widely known as some of the others. And what is an aptamer? Aptamer is nothing but is a short oligonucleotide that can fold into a unique tertiary structure. So, we know what oligonucleotides are. So, these are just sequences of your nucleotides ATGC, and if I arrange them in a certain order such that we have several of them, maybe several of them, and let us say if this sequence continues to 100 nucleotides, then this structure itself can roll around and try to minimize the energy, and in that process it can form a 3-dimensional tertiary structure which could look like something like this. Right, and many other shapes. I mean this is just a random shape that I have drawn. Now again this is fairly small, and we are talking about 100 nucleotides only. So, it is not a huge protein like structure, but it is a small protein like structure, and maybe there is a protein that is complementary to this. So, maybe I have a protein that looks like this, and if that is the case and this aptamer has the sequence complementary or the structure complementary to this protein binding site, then what will happen is this aptamer is going to go ahead and bind to it. So, here is your aptamer, is maybe a protein, and it may not be protein, it could also be lipid, it could also be something else, carbohydrates, it just depends on where you get a hit, but something like that will then have affinity for your target, or I should say protein slash target. So, this is what it relies on, and now the field of DNA synthesis is actually progressed very rapidly, and people have been able to bring the cost down significantly as well as have the throughput very high. So, if I want a sequence which is 100 nuclei, it is not a problem these days, it is fairly cheap, as well as you can have quite a lot of amount being produced in a very small time. So, it is fairly feasible to make lots of aptamers and lots of types of aptamer. So, let us see how then these help. So, as I said, they will recognize a specific target ranging from small organic molecules to proteins to even cells. Sometimes we do not even know what the target is, but we know that it binds to a certain cell, where exactly it is binding to a certain cell may not be known. And so, on the basis of that, people have now come up with a protocol and a method which is called CellX, and this is nothing but it is an systematic evolution of the ligands by exponential enrichment, and it is an evolutional selection method, and we will describe it in a moment as to what it is. And one of the advantage with the aptamer compared to an antibody, let us say we do find an aptamer, and we will go into how we find an aptamer, but let us say we do find an aptamer which is able to bind to a certain target that you are interested in. So, then it offers lots of advantages over antibody. First is aptamer does not have an Fc region. So, as I described that antibodies are large molecules that have an Fc region and a variable region. The basic function of the antibody is to be like a tag for the immune system. So, if an antibody is binding to a certain region or certain protein, that acts as a tag for the immune system to remove that, because that is something that is considered as foreign. And the way the body does that is through this Fc region. This Fc regions has lots of receptors on your immune cells, and once these immune cells bind to this antibody through these Fc region, they will clear it off. So, the last thing that you want is your therapy, that let us say you are putting a particle and you have put an antibody that you are delivering it with it to get detected by the immune system and gets cleared off, then you are losing a lot of your drug, lot of your particle formulation. So, that is one advantage. First, the second is actually it is quite a low molecular weight. So, we are talking about 5 to 10 kDa. Because of that, the diffusion limitations are much lower. So, remember, bigger the size, lesser is the diffusion. So, this can diffuse, this can go through the small gaps, it can find anywhere, it will have a much larger range of penetration into various types of organs and cells. And so, in that regard, this is much better compared to let us say a 100 kDa, 200 kDa protein molecule like an antibody, and that will face this challenge. And I just want to point out that this SELEX method is actually very similar to the phage display library, which is used for selection of some peptides. So, in general, when we are talking about aptamer, you can actually compare them with small peptides. So, small peptides can also have affinity against a particular target, because of the same reason they may acquire a tertiary structure or they may acquire a secondary structure that may have affinity either by the structure or just by the interaction of the peptides with another amino acids on your target. So, this is both of them are similar in terms of their sizes as well as used for targeting, but obviously their backbones are very different, and aptamer do attain a lot of higher tertiary structure compared to peptide, which usually are linear or at max in secondary in nature, secondary structure in nature. So, let us see how does the SELEX work. So, here is your SELEX. So, what you can do is you can have a library of lots and lots of these aptamers in here. So, let us say this is a library of aptamers. And so, you then put your target in it in that library and screen if anything is binding to your target. So, let us say you started with 100,000 aptamers, all different maybe 40 mers or 50 nucleotide long in various permutation combination, and then from there you end up getting maybe let us say 100 of them that is showing some binding to your target. And you can test that through various methods. You can run it through a page gel, and if you see a shift in the molecule, shift in where the protein is coming, you can say that that protein is now bigger because it is bound to some other sequence, in this case the aptamer. Or you can stain for nucleotides on your target itself after washing, and that may show some affinity there. Then you do the in vitro optimization. So, you can take this 100 or maybe these are 10, you can do some binding affinity to figure out which is binding in at a much higher affinity, and you can take them and do your in vitro assays. So, you can see whether you can now target your payload to your cell, what is the mechanism of action. If it does target, if the target stimulation or inhibition whatever you are trying to achieve is happening, then you can basically take it to your in vivo stage, or if it does not happen you can go back to here, you can try to screen with some other library. once that is the case, or if it is happening but if you are not happy with the binding affinity, you can then take that sequence that you know is working in some small amount, and you can try to then mutate it rationally one by one to get a much higher affinity. And then you can go in vivo, you can see whether this works or not, whether it is not binding to any non specifically to any other targets which may then cause toxicity, and if all of this works, then you will essentially have a therapy that is well targeted. And I also want to point out that the DNA technology has progressed quite a bit, and we have now been able to make modifications in your nucleotides. So, here we have LNA, which is a log nucleic acid or peptide nucleic acid, and these are just some variations to nucleotides, and they are actually fairly stable in serum. So, even if you inject this in serum, you find that they do not degrade, they are well tolerated in the body. So, this is one way. The phage library is very similar also, where you are screening for peptides. So, instead of selects library, you will have a phage library, and it is called phage library because we use bacteriophages to which are small viruses of bacteria. So, let us say this is a bacteriophage, and this will display peptides, and you see where these peptides are binding using this phage, and a very similar method will eventually give you a target through peptides also. So, what are some of the shortcomings of aptamer? First is of course that they are negatively charged, these are eventually nucleotides, and so the chances of them interacting with your negatively charged targets. So, let us say my protein is negatively charged. So, if I have a protein that is bearing negative charge, the chances of these aptamer is going to bind to this protein is very low. So, now you have basically reduced your targets to half essentially, because if you assume it is all random distribution, then nearly half of your protein cannot be a target of this. And then the other problem is that it is actually a foreign DNA, because these are DNA that is not what the body is used to. So, it can get recognized by the TLRs, toll like receptors of the immune system, and that may still cause some inflammation as well as rapid clearance. And then lots of aptamers have actually gone through the clinical trials. This is a 2015 data. Some of them have actually been approved for certain treatments. A lot of them you see is actually in quite a advanced clinical trials. There is quite a lot of enthusiasm with use of aptamers as targeting moieties. Along with everything else, there are antibodies which are also in clinics as a targeting moieties, peptides, all of these are fairly widely used. And we will talk about the processes to get clinical approval in our nanotoxicology part of this course. And just very quickly, tumor is again one thing that is widely used for targeting. Basically most of the therapies in research are looking at tumor because it is a major problem, and tumors do upregulate lots of receptors because they need quite a bit of nutrients for their fast growth and generation of new blood vessels. So, as a result there are several molecules that are routinely upregulated and used for tumor targeting. So, transferrin is one, which is a protein that shuttles iron. You have EGF receptor, which is a growth factor receptor. Similarly, folic acid is another one that is required for growth. VEGF is for blood vessel growth. So, all of these as you can see are quite obvious that the tumor will need these to proliferate and grow in size, and so because of this you can then design your particles with these targets displayed that are going to go bind specifically to the tumor cell and get internalized, but not to your healthy cell. So, that way you can then have more specific result compared to what you will get without targeting. However, there are some cons of targeted delivery systems, and some of them are, first of all, that it is important to note that very few targeted delivery systems are actually being used in clinic. So, just an example, of a target delivery system used, and again there are several of them, but just an example is this particular antibody drug conjugate which is used for breast cancer, but there are not many. If you look in the research phase, lot of people are using these targeting moieties, but not all of them are being used in the clinics, and the major reason is it's still debatable whether adding these ligands actually results in any therapeutic administration or therapeutic benefit when you are giving it systemically. So, it is not entirely clear. There are several issues such as once you add a targeting moiety on your particles, you have now increased the size. So, maybe earlier the size was about 100 nanometer, now the size has become 150 nanometer. So, that causes change in pharmacokinetics. You can mask these peptides can get masked by protein adsorption. So, as I said, all foreign material that you are going to inject in the body will come in contact with your proteins that are present, and they may adsorb on the surface, and eventually none of these targeting moieties are actually available. They may change the type of proteins that are adsorbing, they may even denature during their journey throughout the body, and their accessibility is also a question mark. So, that is why there is lots of cons for targeted drug delivery systems. And this is just a laundry list, I just wanted to give it to you. There are lots and lots of clinically approved drug delivery systems. Again, this is probably not the exhaustive list, it is from 2018. So, probably more and more have come out and have been approved, but as you can see, even after all this, all this research we have been able to, as a whole field, been able to actually influence quite a lot of people's life, because we have so many systems that are coming into the market which are showing better efficacy than the free drug. That is why they are approved, and so this is just a laundry list, I wanted to give it to you. Okay, so I think we will stop here, and we will continue this further in the next class. Thank you.